good afternoon and good evening, everybody. We want to welcome all participants and all viewers to this episode 4 of the volume 1 of the Winter Live series by Hadassah International, 50 Shades of Health, a journey into the future of medicine. I'm very excited to be here this evening in Israel, sharing with you the last episode of this year for Hadassah International, and it is my pleasure to introduce and welcome my dear friend and colleague, Guido Melmed, who is going to be taking us through this incredible last episode of the year's journey into the future of medicine. The stage is yours, my friend.
Thank you. It is a great pleasure to be here, and thank you for everyone for joining us for today's special episode. You know, Hadassah doctors are privileged to treat so many complex and challenging medical cases. I want to tell you about one such case, about Adam, who's 60 years old and was diagnosed 3 years ago with renal kidney cancer. Adam was treated with a drug that had a 50% success rate, and for 3 years, he suffered greatly from extensive and ongoing side effects. Now, while I'm telling you about Adam, you may think of any person you know who's fought or is fighting cancer. For Adam, after 3 long years, thankfully, he healed. His treatment worked. Unfortunately, it did not help those who could not wait 3 years. Now, what if a miniature version of Adam's kidney could be produced to test not one drug, but a series of drugs to see which is most effective ahead of time and which had the least side effects? What if we could run this test not over 3 long years, but over a few months, maybe even a few weeks? Does this sound like science fiction? Well, it's not. In fact, it's already underway. And not only is it on its way, you're about to see real such miniature organs and hear more about what is already taking place at the newly established Organoid Center at the Hadassah Hospital in Jerusalem. We're lucky to have with us today a team of scientists that are pioneering. They're building the reality through which this type of medicine will be done. They're paving the way to establish Israel's first organoid center at the Wall Institute for Translational Medicine here at Hadassah. For my first question, I'd like to introduce Dr. Miriam Greenwald, Director of Hadassah's Organoid Center. Dr. Greenwald, thank you for joining us today. Before we take a closer look at the Organoid Center itself, please tell us, how would you explain to someone who's hearing about this for the very first time, what is an organoid and how is it revolutionizing the way we do medicine?
Okay, so to explain what is an organoid, I will first explain what are the tools that we regularly, classically use when we do basic science research. That's the side of research that I come from. I come from the Hadassah Medical School, and we are doing basic science research. And the tools that we are using can be divided into two grossly. The first one is to use what we call in vitro culture, which means that we take one type of cell and we culture it in very artificial conditions as a single monolayer, and we hope that they will behave as they behave in a real organ. And you can understand that is very far from the reality, because a cell in a dish in a completely different environment will not behave as it used to do in a real organ. The second kind of tool that we use in basic science research is animal models, where there we can engineer animals like mice, for example, to have a special mutation or to develop a disease, and then we can study this disease in this specific animal model. But doing so, we kind of forget, or we put aside, that the human body has a much higher level of complexity when you compare it to a mouse, first thing. And second thing, we do not capture the heterogeneity of the human population. And I've been working in basic science research using this tool for many years, but I always show this cartoon illustrating what I just said. When you see a mouse telling a patient, don't worry, I had the same thing, the same disease, and they cured me. And the patient, as you can see, doesn't seem very convinced that he will be cured the same way, because really there is a gap between what we find using these tools in the basic science research and what happens in the real world in the patient because of the limitation of the tools. And you talked about a revolution, and you are right when you use this word, because 10 years ago, a particularly talented gastroenterologist found out, when he was doing his fellowship in the lab of Professor Hans Clevers, that he could, out of a tiny biopsy derived from a patient, you can see Hans Clevers in this video, he could grow, as Professor Clevers shows you, he could grow in a kind of mini organ, recapitulating the heterogeneity of an organ, and more importantly, the 3D interactions of the cells talking continuously, communicating with each other. So now we have this new tool that enable us first to culture the cells in a three-dimensional fashion, so we can recapitulate the 3D interaction between the cells. And second, it is done out of a biopsy derived from a patient, which means that we can capture the heterogeneity of the human population. We can do a mini organ for each patient coming to the hospital.
So you're saying, sorry, you're saying that while we were using the model of a cell in a dish or animal models in the past, now we're actually taking the cell from the patient, producing a miniature organ that belongs to that same patient.
Exactly.
From a genetic perspective, it's compatible to that same patient, and now we can treat or test instead of on the patient in the lab.
In the lab, exactly. And by doing so, we fill this gap between the basic science research and what is happening in the patient, because we have the right 3D construction, we have the right communication between cells, and we have all the genetic background from any patient. So I think that, of course, these organoids have their own limits, but I think that we have now the closest model where we can use that we can use to study in the best way human diseases.
Wow, that's really incredible. Thank you. We'll be coming back to hear more from you about what is taking place at the Hadassah Organoid Center. For my next question, I'd like to introduce Dr. Liron Birenberg-Schwartz, Medical Director of Hadassah's Organoid Center. Dr. Birenberg-Schwartz, thank you for joining us. You're a pediatrician and specifically a pediatric gastroenterologist at Hadassah. Before asking about the particular application that you're exploring, let me just make sure that I understand, because it's truly mind-boggling. We all know about fetal embryonic stem cells, where we take a stem cell from a fetal tissue that has a potential to develop into any organ. But here we're taking cells from an adult. How does that work?
So first of all, hello everybody. Good evening. Chag Sameach. So what happens is that, just like Miriam said, when Sato and Cleavers discovered, the organoids, it all started by discovering that there actually are adult stem cells. Because we all once believed that stem cells is something you have as an embryo or very early in life development. but they were able to show that even in adult organs, there are stem cells. And once they discovered those stem cells, they said, okay, now let's grow them in the lab. Let's see if it works in the lab. So they slowly discovered the right environment in order to make them grow in the lab. And they started with mice, but they were very ambitious people. And when they succeeded with the mice, they said, okay, let's try adults, let's try humans. So they actually started taking biopsies from human tissue. They identified that they also have these adult stem cells. And then they used the same method and gave the right growth factors and the right environment. And yes, they were able to create a mini organ in the lab that was taken from the adults.
So we're looking at cells from adult organs that have regenerative potential. I mean,
Exactly. So we just have to think about it, just like our skin, right? We know that it renews itself all the time. We know the gut renews itself all the time. So there has to be a cell that helps and is responsible for that function, that is responsible for that ability. So those are the adult stem cells. And practically speaking, because of that characteristic, any organ that has stem cells, we can create organoids out of them if we give the right environment. So now we're not stopping in an intestine, which is where everything was discovered. You can actually make organoids from almost every organ these days.
Okay, so now to you. Tell us how you came about to explore and dedicate your professional life to developing organoids.
Yeah, so for me, this is a very important story because I think it kind of modified the way I decided to go as a clinician and now a clinician scientist. So I'll start with a little bit of background because my exposure was in cystic fibrosis research. So just for those who don't know, cystic fibrosis is a genetic disease that involves multiple organs. It can involve the lungs, the GI tract, the pancreas. And these patients many times are very sick, and some of them require lung transplants or liver transplants because of chronic disease. And at a certain point, which was actually 1980s, they discovered that it is caused because of a mutation in a protein that's really important for the balance of electrolytes and fluids in the body. So the treatment of these patients has really improved over the years. And the most significant discovery was the discovery of the CFTR modifying drugs, which are actually drugs that can correct the protein that isn't working well in these patients. But what happened was that they saw that not all the patients respond as we would have expected them to respond. And some patients didn't respond at all to the therapy, even when we thought that they should. So as a group in Utrecht that was led by B actually developed an assay that uses intestinal organoids that are derived from the patient's rectal biopsies. So they took biopsies, which are small pieces of tissue from the CF patients. They created organoids, and they developed this assay that can measure how much this protein is working. And more importantly, can you fix it with drugs and show that the function improves with the drug? So what I did during my fellowship in Toronto was research that was related to cystic fibrosis. So we also used this assay. And one of the very first, it was actually the very first patient that was recruited into that study was a patient with a very severe mutation, which means that this is a patient that has a very significant clinical phenotype. And there are no medications currently available for them. And then while doing our experiments, we tried different combinations of drugs, and one of them worked. And I was sitting in front of the computer, and I was looking at these organoids swelling, which meant that a patient that is not supposed to have functioning protein now has functioning protein because of the drugs that we were able to test on these organoids. And then I understood how significant this research is and how much we can really affect patient outcomes and the way we approach our management of patients. And all I kept on thinking was faster, faster. We have to do this faster. We have to get results. We have to get this out there. And I think that's the point that I just understood, that beyond being a clinician, this is what I want to do. I want to do translational research where what we do in the lab has an influence on the patient and can improve its care.
So here you're actually talking about taking tailored or personalized medicine to the next level, because you have the possibility of screening the range of possible drugs on the organoid, and then understanding which is most suited to the patient.
Yeah. So practically speaking, you take the patient's organoids, which, like you already said, have all the characteristics, their genetic background, and they express all the epithelial cells that they have in that organ. And then you can just plate them. And sometimes you can even play and check up to 120 different drugs and drugs combinations at a time. You can see which one is most effective. And you can even bring it back to the clinician and say, okay, out of the drugs that are available, here's the one that I would try first. And it's not theoretical. This is happening.
And I mentioned maybe a few months, maybe a few weeks. Tell us how long does it take to develop an organoid? I mean,
so practically speaking, once we take a rectal biopsy, and it's really amazing, all you need is one little stem cell. And that one stem cell can actually give you an unlimited amount of organoids that you can keep on expanding, you can freeze it, and then you have an unlimited resource of patient-derived organoids that you can test today, you can test in another month, another 6 months. To your question, it takes the organoids, you can see them developing within a few days. You take that area of the intestine where the stem cells are, and they already start creating within a day or two. But of course, in order to do the experiments, you usually need a larger amount. So depending on the complexity of your experiment, you might have enough within 2 weeks, or you might need a few more weeks, or maybe a month or two in order to make sure that you have enough material or enough organoids to do your experiments. But practically speaking, if the patient is having his surgery, within a few weeks, you would be able to feed back to the clinic and say, here, this is what you might be worth trying.
Just to think about all the suffering that this can really spare patients who go through the trials themselves. It's just
like I said, faster. We have to move faster.
Okay, thank you. I'd like to, for my next question, invite Dr. Alex Gililes-Hillel. Dr. Gililes-Hillel, you're a pediatrician, a pediatric pulmonologist at Hadassah, and a lecturer on pediatrics at the Hadassah Hebrew University Faculty of Medicine in Jerusalem. Thank you for joining us.
Thank you for inviting me. Happy Hanukkah.
Professor Hans Klevels from Holland, who Dr. Greenwald mentioned earlier, and also Dr. Berenberg-Schwartz, is the person who took organoids from a concept that people knew about for years to something that had a wide and has a wide range of applications just a few years ago. Now, you first met Professor Klevels in 2016, so I learned last week, and you're actually collaborating with him and his Organoid Research Center, which is the largest in the world. Now, that's really interesting. Tell us more about this and how you're collaborating.
Yeah, so I was lucky enough to first meet Professor Klevers in 2016 when my mentor and chief pulmonology at the time, Ethan Kerem, decided to go to Utrecht and actually see what they're doing, because there was so much buzz about, organoids, what is it all about, cystic fibrosis research that Liron mentioned. So we decided to go to Utrecht and actually see for ourselves. And this short visit ended up in a very fruitful collaboration. If I have some few minutes to explain what we have learned since, but just to say that as a pulmonologist, I have a special interest in lung disorders, and specifically pediatric pulmonologist, so I study kids with lung disorders. And as you can imagine, there are some more common and there are quite a few rare ones. And what we decided to try, and since this is such a versatile platform that you can basically take any organ from brain to thyroid to pancreas to intestine, and we decided to take airway, lung cells, and to see if we could mimic or recapitulate the disease we see in a patient in this manner to be applicable to lung disease. So what are we talking about? Like, I would like to go back a little bit, a very short introduction to pulmonary lung biology. So we all have airways which lead into our lungs, and the airways begin from the upper airways, which is the nose and the throat, and go down into the lungs, and they're all lined with a specialized cells that have a very specific characteristic. And this is a small hair on each cell. There are hundreds or thousands of hairs on each of those cells, and they're called cilia. And what they do is what you can imagine. They move things out of the lungs to clean them. So that's how our body basically protects itself from things that coming in, whether it's smoke, whether it's viruses, whether it's bacteria, we just push it out. Now, if you have a problem with these cells, with these cilia, you will have problem breathing. For example, if you smoke, those cells will be destroyed. Those hairs will be destroyed. But we wanted to see if we can find a model for a rare disorder called ciliary dyskinesia. Maybe we can see a video of what these cilia look like. Basically, what this disease is that we have, I'll explain maybe for this video, and then I'll talk about the disease. What you're seeing here is an actual organoid that was created in Clever's lab with the samples we provided him from healthy donors and diseased patients. And in a second, we'll see what are those diseased patients. But you can see the hairs for yourself. I think you can appreciate it without a lot of scientific knowledge, what they're doing and how they're moving. And in this video, you see, in the next one, you basically see 4 patients. The one patient on the upper right corner is the one with working cilia. The 2 bottom are those with cilia that don't work. And I'll come back to in a second to explain what is the upper left corner. You can appreciate that the movement is different. So it's not the nice repetitive movement you see on the right. You see a different movement. And the same basically here. So if I don't know if we can move back the video to the last one, just to put it up a little bit more, I wanted to explain about the disease. So the disease is a very rare one. You have a few hundreds of people in Israel. You have a few thousands of people across the world. And basically what it causes, as I said, improper movement of those hairs on your airway cells. Now, the problem with studying this disease is, first of all, you don't have a mouse model. So you cannot create a mouse you can study very well these, because our airway are a little bit different, and at least mice are not a good model for this disease. Now, you might say, let's study them in a dish, but the problem is they survive about 8 days in a dish, and then they die because they need a special surface and interface with air and liquid. Now, if you're able to create a mini organ or mini lung that you can put in a dish and then study it, this would be a very, very interesting area, one, to understand what's going on in this disease. And as you can see here, I told you, the bottom left and right are disease patients from 2 different mutations. The upper left side, there is a movement, but it's not proper movement. And you can see when you create such an organ that this movement is different. And we know that the genetics are different. Sorry, Gideon, you wanted to ask something.
So just to make sure I understand, you're saying this is a rare disease. So this is actually an example of a situation where, without organoids, it's simply more difficult to research because we have less cases to
exactly.
examine, to research. And in so many of these rare diseases, this is a limiting factor and it slows research down. Here we come to a model, a new age really, where we can simply, again, as Liron mentioned, faster and faster. It's transforming our ability to research these rare diseases.
Right. So just using the same example of the ciliary cells, we have in our clinic, and this is one of the largest in Israel at Hadassah for ciliary diseases, we have about 3 to 4 to 7 kids with each mutation. So you can imagine how hard it is to do a clinical trial to test drugs to improve those cilia moving, or even genetic testing if you have very rare mutations. Now, you take in a dish, your sample size increases enormously because you can multiply the same patient again and again and again and use the same patient to study those hair cells, right? Now, going back to smoking, so if we can understand through this rare disease what makes the cilia move better, this would be very, very interesting to a lot of different applications. And again, this would not be possible if we don't understand the basic mechanism of ciliary function through this model.
And on our side here at Hadassah in Jerusalem, you're testing this in the Hadassah Organoid Center as well?
Right. So we started, as I said, the pictures I've shown you were from patients actually from Hadassah that we collaborated with Clever's lab, and we're currently in process of publishing these data because we think it's very exciting that we were able to create an organ of real ciliary movement and to show that it really corresponds to the patient, not just a general ciliary. And we are planning now to begin doing the same with samples from Hadassah's patients. So definitely, yes.
Wow. Okay, I would like at this point in time to come back to you, Dr. Greenwald. Everyone wants to know where we're going and how long it's going to take to get there. Now, you are bringing this technology, all of you, to Israel, making Hadassah the first organoid center in Israel. Tell us what your plan is.
So just now what we are doing is establishing a bank of organoid. What I mean by a bank is a huge stock of organoids that we have grown from as many different organs that we can collect from patients, Israeli patients, and from different kind of individuals, men, women, children, different diseases, different organs, trying to cover as many diseases and as many different genotypes, different genetic backgrounds that we can. And we have to point out that in Israel, we are blessed with a so heterogeneous population. We have people coming from Russia, from Ethiopia, from Europe, from America, so we can cover really all different genetic backgrounds. So this is one of our first goals, to establish this organoid bank.
Tell us, sorry, if I may, tell us what we're seeing here. These are actual organoids.
Yeah, here we have just finished to develop a certain type of organoid. Here it's an organoid derived from a biopsy of a cystic fibrosis patient. And we just can freeze it in a -80 degrees, and we will be able to thaw it on demand to test any drug, to do also basic science research, and to do whatever we want to test on this organoid, and not directly in the patient. And as you mentioned before, it saves so much pain, so much time, so much effort, just by freezing and these small organoids.
So you mentioned the organoid bank. Today, we know how to transplant the heart of, or the medical world knows how to transplant the heart or the kidney of Mr. Cohen and Mr. Levy. are we looking at a reality where we'll have a bank of organoids that
Yeah.
we will be able to inject into an organ that needs healing? Or how far are we from that? Is it already happening?
So it sounds like science fiction that we could grow a mini organ, freeze it, and when Mr. Cohen comes because he has a problem in his gut, for example, we though his organoid, grow it a little bit in the dish and then reimplant it. So it sounds science fiction, but it's really happening now, at least in mice. In Japan, just in the last month, they have initiated this new experiment where they have grown human gut organoids, and they can retransplant these human in mice that have been engineered to not be able to reject a human tissue. And these mice are suffering from a very common bowel disease, and they have done that already mice to mice, and it works. And now they are doing human to mice, and they hope that these little mini organs will flow into the gut, find the lesions that are in different parts of the gut, and just integrate to repair the non-functional gut part of the gut.
So this futuristic reality
Yeah.
sounds closer than ever.
Yeah.
I just want to yes, please.
Yeah, but meanwhile, what we are going to do with this bank of organoid, it is a platform that will be used not for transplantation, because it will be in the next few years, but for answering basic scientific questions, and also for clinicians. If for example, an oncologist wants to test a drug or a sequence of drugs that he wants to treat his patients, we will be able to take out the organoid from a tumor of this particular patient, and test the drugs and test the sequence of the drugs he can use. And as Leon mentioned, come back to the clinician and tell him what drug he could use to have the most beneficial effect.
Okay, well, we are approaching the end of this episode. Actually, I would just want to ask one last question regarding the Organoid Center at Hadassah. You know, it's the first of its kind in Israel. Are you working or are you interacting with scientists outside of Hadassah to serve the country's wealth of potential in research? How are you going about this?
So this technology has been rapidly adopted by different labs around the world, and also in Israel. Some particular labs have imported this technology to answer their specific question. What we are doing at Hadassah is to develop this platform to serve all the scientists, both from the clinical side and from the more basic science research, to answer this question. And our center was born just 2 months ago, and we are already approached by many scientists of both sides. And we are initiating, for example, a very important study to check the interaction of the bacteria that we all have in our gut with the cells that are lying in the gut, to understand how bacteria can push the cells to become tumor cells. And we do that with a very famous lab in the Hebrew University, led by Professor Inon Ben Neria. And I think that this is one of the projects that he couldn't do without organoids, without this technology. And we are doing that right now in the center.
So there's a tremendous collaborative potential, generally speaking. This has been absolutely fascinating and enlightening, and a beacon of hope, really, for so many people. I want to thank you, Dr. Greenwald and Dr. Birenberg-Schwartz and Dr. Gilles Hillel. Thank you for what you're doing, first and foremost, and thank you for making the time and being here today with us. There's so much more to talk about. Literally, we could spend a couple of hours doing so. Unfortunately, the time is up. I want to also thank everyone for joining us today. And I also, before wrapping up, would like to thank Hadassah International for initiating this series. I also want to acknowledge the Wall Legacy for establishing the Wall Institute, where this activity is taking place, and the Hadassah Women Zionist Organization of America, whose generous support has made it possible to establish the Organoid Center at Hadassah. Before wrapping up, I'd like to introduce Robert Dorfman, President of Hadassah International, directly from Hong Kong, and to invite back Executive Director of Hadassah International, Jorge Diener, for closing remarks of Volume 1 of the Winter Live series. Thank you.
Thank you very much, Gidon. I want to take this opportunity just to thank Dr. Miriam Grunwald, Dr. Liron Birenberg-Schwartz, and Dr. Alex Hillel, who have been with us today for a fascinating presentation. Thank you very much for the great work that you do. This is the 4th episode of our Winter Live series. We take a break as we go into the new year, but we will resume again in early January and continue through February. I want to thank our outstanding team in Israel, led by our Executive Director, Jorge Diener, for all the work in preparing for this series. Also, a big thank you to Gidon Melmed for having been such a wonderful moderator today and on a number of other occasions. And we look forward to continuing working with you on this, Gidon. Thank you very, very much. Also, our extraordinary medical professionals, 3 of whom you just saw at Hadassah, for sharing their wisdom, their important work with us through our Winter Live series. Also, a very big thank you to our global Hadassah community for your participation in these calls. This is a time of great concern over what's happening in the world in terms of health, and the desire for knowledge and understanding of what developments are taking place has never been at a higher level. So thank you to all of you for being with us, and we look forward to having you with us as we go into the new year for the continuation of our Winter Live series. Thank you very much, and Jorge, let me pass it over to you.
Thank you, Rob. Thank you, Gidon. Thanks, everybody. Thanks, all the doctors. It's I'm very moved by closing this volume 1 of our Winter Live series, which actually is also the last Hadassah International virtual event of 2020. And I think it's very symbolic that we close this series, or this first part of the winter series and our last event of the year in the middle of this season. You know, we are in a season that some people call it the season of miracles, and others might call it the season of lights. And I think I want to rename it, and I think this is the season of lights at the end of the tunnel, because this is the moment that we all look at when the darkness will end, when this period that has been so extraordinary for everybody will finally finish, and we will see the light at the end. And I think that one of the things that we try to do here at Hadassah International during the whole entire year of 2020 is to bring that light of hope during the worst times of COVID in the early months of March, April, May, June, and then with our summer program, our summer summit, when we started looking at what is happening at the end, what's happening after, you know, we finish COVID, what are the other diseases, what are the discoveries happening at Hadassah. And this specific Winter Live series that take us into the journey to the future of medicine, take us far away, take us not to the light that we are going to see. We all know we are going to see the light ending out of this pandemic period in 6 months. And we will remember the year, or we will probably want to forget this year. But there will be things that we will remember, and for me and for many of our friends all over the world, and our fan club, and who are all the time following our episodes. I think this is the kind of things that we will remember, and we'll be grateful for the doctors that have been here, not just sharing what they do, but also sharing what they dream. And I think this is so much about what Hadassah, HMO, International, HWCA, the Adasa Women Science Organization of America. This is so much what we're all about. It's not about only what we do, but it's about what we dream day by day. And that's the way that we made the difference in the world, and we keep making the difference in every one of our actions. Those 8 candles that both Rob and myself have in our background with the Hanukkah are just a symbol of how we can accomplish everything if we believe not just in miracles, also if we believe that there are lights that we can light in ourselves when we're lighting the world by healing step by step. I want to thank you all, and I want to make sure that you see in the next 2 minutes what was during this Winter Life series and what is coming in January and February. Please stay with us for the next 2 minutes to see what's coming next. Shades of Health, a journey into the future of medicine.
What will medical center look in the future?
Computer science,
AI and machine learning,
semi-digital lenses,
revolution in medicine,
a multi-machine room,
combining optics and nanotechnologies.